Hi, I'm Mike Fricker and I'm a research fellow based at the University of Newcastle and Hunter Medical Research Institute in New South Wales, Australia. Today, on behalf of my co-authors, I'm going to present a brief summary of our recently published manuscript titled Circulating Mast Cell Progenitors are Depleted by Benralizumab in Severe Asthma. So we all know that mast cells are key therapeutic targets in severe asthma. They're dysregulated in terms of their number and function across airway compartments, especially in severe asthma. The origin of these increased lung mast cells in asthma remains somewhat unclear. They could be derived through proliferation of local tissue mast cells or via recruitment and differentiation of systemic progenitors. Recently, circulating mast cell progenitors were immunologically defined in humans, and these have been shown to be increased in number during pollen season in mild atopic asthma. The clinical associations of these mast cell progenitors in severe asthma has not been studied, and the impact of severe asthma monoclonal antibody therapies on mast cell progenitors is unknown, although these cells have been shown to be IgE and IL-5 responsive in vitro. To address this knowledge gap, we performed an observational clinical study. In a cross-sectional study, we recruited 80 adult participants with severe asthma, comparing outcomes across non-eosinophilic, eosinophilic, and mepolizumab-treated severe asthma. And of the 32 participants with eosinophilic asthma in the cross-sectional study, we followed up 30, 20 of whom commenced mepolizumab treatment, and 10 of whom commenced benralizumab treatment after the first study visit, which we termed V1. These participants were followed up at 8 weeks, which is V2, and 24 or more weeks, V3, following commencement of treatment. The details of this cohort were published last year in Allergy, so I please encourage you to check that out. We measured mast cell progenitors in freshly isolated peripheral blood mononuclear cells using previously published flow cytometry criteria. And MCPs were identified as lineage negative, CD34 positive, CD117 positive, and Fc epsilon receptor positive progenitors. In our longitudinal study, we observed that benralizumab almost completely depleted circulating mast cell progenitors, while mepolizumab had no impact on their abundance. We found that a subset of mast cell progenitors had detectable surface IL-5 receptor expression in all study samples, while expression of IL-4 receptor alpha, which is the target of dupilumab, was absent. Benralizumab completely depleted MCPs that were IL-5 receptor positive, while impacts on IL-5 receptor negative MCPs were less emphatic, consistent with its mechanism of action. To extend our findings, we sought to develop a gene expression signature that could report mast cell progenitor abundance in blood or PBMC bulk transcriptomic datasets. To do this, we identified expression of genes that were restricted to mast cell progenitors among some 836,000 single blood cell transcriptomes. In this dataset, mast cell progenitor identity was confirmed through high surface protein expression of CD34, CD117, and Fc epsilon receptor, which is consistent with the flow cytometry strategy. We found 6 genes were mast cell progenitor restricted, which included histidine decarboxylase, which is shown here. The other 5 genes were also mast cell relevant, including mast cell proteases, IgE receptor subunits, and the prostaglandin D synthase. We examined mast cell progenitor gene expression pre and post 4 months of benralizumab treatment in 41 severe asthma patients in a previously published dataset. And we found that all those genes that were detected in this dataset were depleted after treatment, which validated our flow cytometry observations. We went on to use our mast cell progenitor gene signature to explore their clinical significance in severe asthma in other published datasets. Mast cell progenitor gene expression did not differ between healthy, mild to moderate, and severe asthma groups in both UBIOPRED and IMSA cohorts. Furthermore, mast cell progenitor gene expression was unaltered by omalizumab treatment in 45 severe asthma patients, and was also unaltered by dupilumab treatment after 3 or 4 months in 2 separate atopic dermatitis studies. So depletion of mast cell progenitors appears to be specific to benralizumab among the T2 biologics that are used in severe asthma populations. In our longitudinal flow cytometry cohort, we found that lower mast cell progenitor abundance after 6 months of MEPO or benralizumab treatment was associated with greater improvement in asthma control, and this suggests that there may be some clinical significance associated with mast cell depletion in severe eosinophilic asthma. So I'd like to thank you for your attention, and if you do have any questions about the study, please check out the full article on the Allergy website and contact me by email if you have any questions. Thanks.